Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Kinds of systems that...
Your mic's not on.
Mic's not on.
Yeah, it's your top switch comes up.
How's that? Okay? Okay. And so I'm going to talk today about a facility at the University of Texas in the geology department that has a high-resolution X-ray scanner capable of scanning both small and medium and somewhat larger size objects at very high resolution with lots of X-rays and photons that result from those X-rays. But my interests are in the area of morphology and how morphology helps us explain and understand what happens in the world around us. Historically, the field of morphology and anatomy was one that was associated with illustration. And the great anatomists of previous centuries were always illustrators. But today, we have the advantage of being able to capture morphological information digitally. And that digital revolution has had an enormous impact on morphology and anatomy. And specifically, although 2D scanners and cameras collect digital information about morphology, you can take a picture of a specimen or an animal or plant. What has really made a difference is the ability to capture these same organisms, these same features in 3 dimensions. The scanner that we have is comparable to a medical scanner, except that rather than the scanner moving around you as you are inside a very dark tube, the organism or the specimen itself rotates. This is obviously works only on things that are already dead and on a wide variety of objects with varying degrees of density. We have very high resolution systems capable of resolutions down to a few microns. The workflow here, the data flow here is that the actual scan produces something called a sinogram, which is the actual image shot from the detectors as the specimen rotates around, frequently 1,200, 1,600, or 2,000 shots as the specimen rotates. Those are reconstructed using a back projection algorithm into actual slices. A typical data set for us is anywhere from 500 to 2,000 1,000 by 1,000 pixel slices, 16-bit depth, color depth. And so we get data sets anywhere from 1 to 4 gigabytes in size. Our detector capabilities are probably going to be doubling in the near future, so we will get 2K by 2K resolution. Now we're starting to get quite sizable data sets that are hard to actually manage for normal applications. What we do with those data are various. Primarily for most of the applications that we're doing, and we started as a digital library project funded by the National Science Foundation. My project did. I'll explain more about some of the other applications, in which our goal was to scan and disseminate information about some of the rarest and most important natural history specimens on Earth. And so our immediate goal was to scan those specimens and use visualization software on those to produce imagery and animations that let people actually see those objects in 3 dimensions. Those products are also part of our workflow. We produce animation frames and movies that result from those using a variety of proprietary applications. And so I'm going to show you a little bit about, I'm going to go over to a browser and show you the facility itself, the digital library that has been the mainstay of our dissemination output for the past 6 years, a little bit about UTCT, which is our recent digital archive for disseminating the raw data, and I'll briefly mention some possibilities where we might be going next year. So let's go over here and go to a browser. The facility itself is also NSF-supported multi-user facility that is located in the geology department. It is entirely a soft money operation, operates on a combination of NSF funds and user fees. We're probably the most successful, at least NSF keeps telling us, multi-user facility that they support, in part because we have so many outside collaborators. We end up with 50 to 150 outside collaborations or clients a year who bring things in to scan. And the types of things that they bring in to scan are just, almost anything that you can imagine that might have something on the inside of it. And so many of these are geological samples that I don't really know a lot about. These are meteorites, for example, here, in which the important thing was the vesicles that are located inside of them. We have this is an a drill core in the bottom of a open pit gold mine in Colorado that they were not sure whether there were further gold samples there, so they took a core. In this case, you're seeing a plane inside of inside of this core that, and actually those little red flecks are actually gold. And so that's one of the kinds of geological samples that we do. We also do a variety of engineering samples. These are asphalt cores. Asphalt is a big business, a remarkably big business. Everybody wants smart asphalt that lasts and has the right amount of permeability and does all these things that make our roads better. And it turns out that's big science. And so when they make an asphalt core, once they've done that, they have no idea what's on the inside, how they ever fit together, and so forth. And so they bring this to us, and we scan a lot of asphalt cores. This is an interesting book that was we scanned as just a demonstration of showing that you can, in fact, look inside of these books. It's probably going to take too long, so I won't do that. But you can actually read the book as you scan through it. And so, at any rate, so that's the facility itself. The digital library that I'm associated with is the Digital Morph project. We have approximately 500 biological objects that we have scanned, everything from flowers to fossils, dinosaurs, fish, birds. Our most recent scan thing here is a baby and an adult skull of an emperor penguin, the one from the most recent movie. So these are specimens that for which we have made animations. I have one loaded here. So this is a 3D rendering, volume rendering, using proprietary visualization software for which we have, over the course of probably 3 or 4 hours, shot a 90-frame animation and saved that out as a QuickTime movie. We actually only provide on the site these web-sized versions of these movies. We make these in much larger, richer formats, but the distribution of those is more difficult because of their size. So typical movies for us at full size range from anywhere from 10 to 50 megabytes. The amount of detail in these, this is reduced. This is actually about twice again as big as my screen. Is significant. And these are the rendered views of the data. We also offer the capability of looking at the raw slice data itself. This is the skull of the specimen, the adult specimen of the emperor penguin. Now, one of the things that's a little hard to get your mind around with these data that differs from what you do when you snap a picture is that these are true geometrically correct data. So we have this skull now rendered as a movie, but we also have those data correctly proportioned in 3D space. And so this slice viewer that you see here, which is a fairly low technology Java applet, okay, does in fact retain at all times the correct geometry of the specimen. So I can ask a simple question like, how long is this skull? And see that it's 187.22 millimeters, and that's a correct number. And so part of our workflow issue is that we have this metadata associated with the voxels. The voxels have a geometry, and whenever we carry those into the next step, it's important for us to manage that particular aspect of the information. And so, this has been our primary output format for the last 6 years. It's great. It's very popular. It's won, Scientific American awards. It was one of Yahoo's top 25 science sites a couple years ago. We get a lot of interest from educational audiences, a lot of outreach types of purposes, but it's a little bit, maybe a lot low on resolution for scientific applications. The full resolution data sets, the full size animations, when scientists are looking at individual features that they're interested in, and I have a movie here which has been segmented. So we have now got, somebody's actually gone into the individual, the voxels and annotated them as to what parts they were. This is a larger set of those. There's actually a subset of these. It's hierarchically organized. And so each one of those is given a different transfer function, and the resulting voxels are colored in an animation. This type of data, and even at higher resolutions, are what the scientific community requires. And so we have, a year or so ago, started a project to disseminate these information and provide tools for examining the full-size data sets. And that is something we call UTCT. We have only a small number of data sets on hand. After I'm going to show you some demonstration of this, I'll talk a little bit more about it when I move back to PowerPoint. The architecture of this is that we are storing the information in SQL Server, the individual images. Outside of that was a challenge. The processes of loading those data and managing them has proved to be something that took us a while to get worked out. And we're still not yet having, we still, as of this point, still don't have a completely smooth workflow process for moving the data from our lab into this system. But we've got many of the pieces in place, and much of the functionality is starting to get there. What we offer right now are a couple different reasonably straightforward visualization system tools. We have a light table viewer that lets you look at the data in a light table view, needless to say. You can pop on individual frames and look at those. This is really good for sort of getting a feeling for the data, what's involved in that. If that wasn't a big enough picture, you can change the scale on these things and look at slightly larger versions to see what it is that's present in the data. It's a very quick way to go through the data. We also have another applet on this site that, in fact, needs to have a fair amount more work done to it. And this in case we're looking at a mouse. And so put it in a place where there is something there. Again, offering views through 3 primary cardinal orientations. Okay, so let me go back to a slightly better description of the site and what's happening. Okay, so in theory, what we'd be moving into this is 10 to 15 data sets a week if we actually moved all of our data flow out of the lab into this system. And that would be a lot of work under our current system, in which involves FTPing stuff and typing stuff into scripts and so forth. And so we are very definitely interested in improving that process. We as part of it, collect the metadata that keeps track of the geometry, the information about the imagery, about the data set, ancillary imagery. The data are maintained in SQL Server. The server itself that you just saw is Apache with PHP scripts. One of the important steps in this is that we do, the data set comes out as TIFF slices through the data as it was scanned, but I showed you in each of these cases views through the data in the other cardinal orientations, horizontally and sagittally, and those images have to be produced ahead of time. Producing them on the fly would be too slow, so we do those in a script using an IDL, and we load the data with Java and a set of stored procedures that ultimately call textcopy.exe to individually load each image one at a time. And so we have an architecture. And so the workflow as it stands now is we FTP these things over. We do an interface thing. We start the Java application that loads the data. It converts the data from 16-bit to 8-bit. We maintain both 8 and 16-bit versions of the data. We can make a JPEG version of the data. All those slices get loaded into the database and sort of magically appear on the website. And so that's our current where we are, okay? But I want to also spend some time talking about the process that gives rise to this being an important aspect of a larger enterprise, and that is the scientific enterprise associated with the tree of life, the history of life on Earth, and current efforts to understand that history of life and to talk about the processes that are involved in that and to look at the features on organisms that are manifested by our understanding of that tree of life. And so I'm going to go briefly through, trying to keep going fast here, the general enterprise of assembling the tree of life, one that starts with biological specimens in museums and herbariums, the collection of data, the collection of specimens and data associated with them, the creation of taxonomies that keep track of the names associated with these organisms. These are all done by systematists. All of those first steps are done by systematists who work in museums, who work on groups of organisms. The process of creating trees, or recreating the history of life, is one in which we look for characters that tell us information about organisms and the features they possess. We do that two ways. We can do it with sequence data. Lots of people are doing it with sequence data. We can do it with morphological data. Even when we do it with sequence data, we're interested in the changes that happened on organisms, the morphological changes that happen on organisms that we can see with our eyes. And so the process of mapping morphology onto trees or creating trees, phylogenetic trees with morphology, are much in the same in terms of the informatics infrastructure. So briefly, I've outlined here a diagram that provides some of the steps that, and I'm going to tell you where the steps that we're involved in after I go through this. And so natural history museums collect specimens. In the case of morphology, cameras and scanners collect information collect documentation about features. And they may be snapshots, they may be hand illustrations, or whatever they are. Those, of course, all have to be managed in the standard way. If it's digital cameras and 2D images, they can go right to your desktop for observational purposes. If they are 3D data scanners, they would have to be visualized in the fashion that I just talked about. The scientist's desktop and what happens there, I'm going to come back to that in just a minute. But the process that most scientists undergo is that they'll sit there and they'll take a collection of organisms, sunflowers, okay? And there may be 400 species of sunflowers in the world. I don't actually know that. I'm not a botanist. I don't know why I picked a botanical example. But they will go to museums. They will look at images of sunflowers. They will hold them in their hands. They will snap pictures. They will make drawings. If they 3D object, they'll scan them, scan a seed, and look on the inside of it. And they will take those 400, for each of those 400 taxa, they will collect characters, morphological features. This is something that has happened forever, okay? Darwin noted features of organisms and how they vary between taxa. So he recorded characters and he recorded states of those characters for individual taxa. Now, that process has happened time and again, the same organisms many times, and one of the sort of weaknesses of the entire enterprise is that the concept of characters that are collected on organisms is one that has been historically done time and again with scientists individually coming up with those characters and the names for those characters. We have pretty good concepts for the basic structural elements of the features of a few organisms, the mouse, the human, not much beyond that, actually. Most organisms on Earth have unspecified vocabularies for the features that characterize their morphology. So the skeletal elements of a goldfish are ones that for which there are published papers, but there are no standards for what each of those elements is called. Humans have obviously had a lot of work done on them. Mice have had a lot of work done on them, but even the mouse ontology for structural features is incomplete. And so the process of what's happening on this scientist's desktop is that he's looking at these features. He's looking at the features of the sunflower. He's looking at the features of a mouse or a goldfish, and looking for variation amongst the taxa in hand. And when he does that, he or she records those in a matrix. And that matrix will have ultimately a code, a 0 or 1, that corresponds to some state of some feature, okay? And so in this particular case, we're looking at some small critter that I'm not sure what it is. And so I still don't know what it is. And has decided that there are two states of that, okay? And in this particular case, has documented them. This is a desktop application known as Mesquite, in which it does have the capability of storing this trait that has been, it characterizes the individual states for this particular character. The base of the claw tuft, okay, in taxon Coccolodtes and a variety of other taxa here also have been either recorded or not. Now, the feature that I want to talk about here is that this is done effectively in isolation, okay? This is a great application. It takes these images and stores them, sticks them on your desktop, and that's the end of it, okay? This person's going to collect all of these, and the next time they look at it, it'll still be there. But there's nothing in our enterprise whatsoever for the maintenance of this information. The reduced set matrix, the ones and zeros with the names of the columns and the rows, is stored. It's published in a paper publication. There's at least opportunities to put it in public repositories, but the information about what constituted the zeros and ones is gone forever except as it occurs in a paper publication. So, Dan Maranker and I are engaged in a recently funded project from the National Science Foundation to try to improve the infrastructure associated with this process. So this is a process that starts with the collection of morphological information, be it from a scanner or digital camera, or hand illustration, or photocopy out of a book, and takes that, looks at it, decides how to break up that information into characters and character states, documents those in a desktop application, and now we're going to move to a community infrastructure in which we have individual desktop clients, much like what we just saw, but connected into collaborative projects where individual researchers are able to share those results real time with their collaborators, checking in and out rows, columns, and cells of those particular matrices, and that the words that correspond to those characters and character states are ones that are stored in community ontologies that are resolved through LSIDs. And so that enterprise is one that we are at the absolute infancy of. In fact, we're at the infancy of so much that this is about all that exists for it right now, is a diagram that is a conceptual view of how our community might interact in a fashion that corresponds to what a lot of other communities are already engaged in. And so we envision this as being one in which we prevent an awful lot of redundancy, that we improve the accuracy of work that's done, because when we use a word, we can see what it means by looking it up in an ontology. The ontologies will have associated with them documentation in the form of digital imagery and words and annotations, and the communities that come after us will have the benefit of being able to connect the results of our scientific research in terms of trees and transformation of characters on those trees with the underlying data that gave rise to them. And so I will stop with that. Questions? Yes?
I actually work with biologists who work with systematists
Absolutely.
So what would you do in case there are doubts about whether this is a innate or a learned response?
Right. Okay. So we have, there's two pieces of information here. One are the characters that are associated with the observations that the systematists make. The other ones are their results. Their results are trees and the mapping of characters on those trees. Part of this larger infrastructure that's at its infancy is the ability to store those trees in community databases. Again, up until literally today, those trees are stored simply in paper journals. And if you want to look at them, you literally hold them up and make a Xerox of them and put it on your screen and send it to somebody else to talk about. I mean, really, really no ability to talk about these electronically. So what we envision is that there's no reason that we have to assume a particular classification, a particular topology of trees to talk about the characters. Oh, okay. Okay. Yes, change things out. And so for us, that disagreement about topologies is not really the issue. Yes, we fight about those, okay, but the fact of the matter is that the identification of individual organisms, what they fit, how they fit into the tree of life, is one that's never going to be fully resolved. I mean, it's nice to think that if we had full sequences on every organism on Earth, we'd actually know what the history of life was. But the truth is, we would still not know. We have complete sequences on chimps and humans, close on most of the other primates, and we still aren't positive. Those are things that are still. We don't have high degrees of certainty on things that are the most studied on Earth, okay? So it's hard to imagine that we're ever going to have the answer for the tree of life. So we're always going to have those discussions. But what we also have are the observations that we make on those organisms. You know, how many fingers they have, how many toes they have, what color hair they have. And so those are things that we can talk about and talk about the impact of those things given different topologies of the tree of life. This is the basis of discussion. It'll be much more educated discussion if those can be mapped on there. Sometimes those mappings are what actually helps us understand whether or not one of those topologies is more likely or not.